This video is a recording of the 5th webinar in the SGD webinar series, titled Exploring Expression Datasets and Co-Expressed Genes with SPELL. Please enjoy. Hello and welcome everyone to the 5th webinar in the SGD webinar series. My name is Kevin McPherson. I'm an assistant biocurator at the Saccharomyces Genome Database, SGD, and today I'm going to demonstrate how to use the expression dataset search engine, SPELL. Before we begin, please feel free to ask questions anytime throughout the webinar. Just click on the Q&A button on the right-hand side of the Blue Genes page, as shown by the red arrow, and the SGD team will answer your questions as they come in. We'll also be having a Q&A session at the end, so please be sure to stick around for that if you have any further questions. So what is SPELL? SPELL is a web-based search engine for large-scale gene expression microarray compendia that was developed by the Troy and Skya lab at Princeton University. Given a set of query genes, SPELL identifies expression datasets from published microarray experiments and ranks them according to their relevance to the query. SPELL also identifies genes from these datasets that have similar expression profiles to the query set. In essence, SPELL is a discovery tool that connects your genes of interest to informative expression datasets and co-expressed genes. Results from SPELL queries are ordered into a matrix like the one shown here, which can be clicked on and explored for further analysis. To access SPELL, open the SGD homepage at yeastgenome.org, open the Function tab, and click on Expression, as shown by the red arrow. Or, if you're already on a locus summary page at SGD, just open the Expression tab. This will take you to the gene expression page on SGD, which has a link to the SPELL tool for that gene, as well as a histogram that is created from the same data we use to populate SPELL. Lastly, you could just type in the URL, spell.yeastgenome.org. So let's start the tutorial by going over how to run a multi-gene query. Here's the SPELL entry page, again at spell.yeastgenome.org. To run a query with multiple genes, enter or paste your genes of interest in the search box and click on Search. Note that SPELL will accept either systematic or standard gene names, and that separating gene names with commas is optional. Once the search is completed, SPELL displays the results as a heat map. The query genes we entered are shown here, while datasets relevant to these genes are shown at the top. SPELL determines the relevance of these datasets based off of co-expression levels for your query genes. In other words, datasets where query genes are highly co-expressed are deemed more relevant than datasets where query genes are less co-expressed in comparison. By default, only the top 10 most relevant datasets are shown. To display lower-ranking datasets instead, you can select them in intervals of 10 in the Datasets to View pull-down menu. As a side note, it's worth mentioning that if you run a SPELL query with only one gene, then all the datasets will be considered equally relevant because there are no co-expression levels to compare. Click on any of the dataset titles to be taken to a page that provides more information on the publication behind it. Note that each dataset has one or more tags, shown here, that indicates the general biological topic investigated by its associated publication. In this example, we see that cell cycle-related datasets are identified as being the most informative. If you'd like to filter for datasets that have a particular tag, expand the Dataset Tags list here. Select the tags that you wish to filter for, and then click on Update. Note that SPELL does not recalculate the results when filters are applied. It only changes which datasets are presented to you. As mentioned before, query genes and their expression profiles are displayed here. Following them are other genes with similar expression profiles, ranked by their level of co-expression across all the datasets in SPELL. To examine a gene's expression profile for a particular dataset in more detail, just click on the appropriate patch in the heat map. This will open a window that displays the number values associated with the expression data and the publication it belongs to. If you'd like to add any of the enriched genes to your actual query, or remove a gene from your query set, just check or uncheck the genes as appropriate, and then click Update. Spell also uses all of the genes in your results to produce an enrichment table of gene ontology terms, or GO terms for short, at the bottom of the page. Briefly, gene ontology terms are standardized phrases in a hierarchical structure that describe the biology and function of gene products. The enrichment produced by Spell indicates which gene ontology terms are highly enriched with the results of your query. In this case, terms such as chromosome and DNA replication are listed among the top. Spell also lists the calculated p-value and the percentage of genes within the query and across the genome that share this gene ontology term. Click on any of the gene ontology terms if you'd like to find out more information on that term on the gene ontology website. Lastly, if we return to the top of the page, there is an additional display options menu that has two more important settings. Mapping method, to change how Spell displays single and dual channel arrays, and color scheme, for switching the color of the heat map from red-green to blue-yellow. That pretty much covers how to run multi-gene queries in Spell. To wrap up this webinar, let me demonstrate one of the practical ways that you can use this tool: predicting members of a biological pathway, such as glycolysis, based on co-expression levels. Recall that Spell identifies genes with similar expression profiles to your query set. Because of this, running a multi-gene query in Spell, where your query genes are all related to a particular biological pathway, might identify novel members of that pathway based on co-expression. Let's use glycolysis to test this idea. If we enter in an incomplete set of glycolysis genes into a Spell query, will Spell identify it's the missing members? The first thing to do is to obtain a set of genes related to glycolysis. This can be done easily on the SGD website. I'll just look up glycolysis in the search box. Click on the genes category, since I'm looking for genes, and scroll down to the bottom of the page to expand the biological process options. Then I'll click on glycolytic process, and now I've got 19 genes that are annotated to glycolytic process and also have the word glycolysis somewhere in their description. To download these genes so that I can import them into Spell, I will click on Wrapped and then on Download. Returning to Spell, paste the downloaded genes into the search box. In this example, I'm going to delete the first gene in this list, FBA1, to see if Spell will identify it as an informative gene for the entire glycolysis query. And there we have it. Spell identified FBA1 as a highly relevant gene, based on its co-expression level with the glycolysis query. Other genes enriched to the top are also relevant to glycolysis or its byproducts. ADH1 is alcohol dehydrogenase, which catalyzes the last step in glycolytic fermentation to ethanol, while PDC1 is pyruvate decarboxylase, which decarboxylates pyruvate from glycolysis into acetaldehyde. The final thing I'd like to mention is that if you'd like to find out more about the algorithm behind Spell, the version of Spell hosted by SGD, and other implementation details, just see About the website here on the left's menu. This concludes our webinar. Thank you for coming, and please be sure to keep an eye out for our follow-up email for more information about Spell, our next webinar, and the link to our YouTube channel, where today's webinar will be uploaded. So thank you again, and let's go ahead and start our Q&A session. Thank you for watching the SGD webinar series. If you have any questions or comments, please contact us at sgd-helpdesk@lists.stanford.edu.